Hello, everyone, and welcome to today's webinar, a novel method for quantitative Western blotting that eliminates errors from the use of housekeeping proteins. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want and any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now present today's speaker, Dr. Alok Tomar, Product Manager, Protein and Cell Analysis at Thermo Fisher Scientific. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Alok, you may now begin your presentation. Welcome.
So thank you for that introduction. So today we are going to talk about an exciting webinar that we are doing, which is titled A Novel Method for Quantitative Western Blotting that Eliminates Errors from the Use of Housekeeping Proteins. I am the product manager who is based out of Carlsbad in sunny California. And Carlsbad is the site where this No-Stain reagent that we are going to talk about is made, along with our precast gels. So this is basically a brief agenda for today's webinar. So we will review the need for protein normalization in Western blotting. We will review data generated using multiple cell lines and popular target proteins with regards to protein quantification using two separate methods, one with using housekeeping proteins and other with total protein normalization. And finally, we will review the benefits of using No-Stain Protein Labeling Reagent for total protein normalization. So the most common method currently being used for protein normalization with Western blotting is using the housekeeping proteins. The table here on the right lists the most commonly used housekeeping proteins that include proteins like actin, GAPDH, tubulin, etc. The housekeeping proteins, they're assumed to be expressed constitutively at the same level across experiments. However, recent studies have shown that expression of housekeeping proteins can change across different cell types and biological conditions. Therefore, scientific publishers and funding agencies, they now require other forms of normalization or validation of normalization controls to ensure reproducibility and accuracy of quantitative Western blotting results. So this slide, it demonstrates really one of the biggest issues with using housekeeping proteins as loading control in quantitative Western blotting. So in this particular experiment, HeLa cell lysate was loaded with increasing concentrations from 10 microgram to 50 microgram. The lysate was transferred onto a membrane and probed with one of the most commonly used housekeeping proteins, so beta-actin, GAPDH, and alpha-tubulin. Now, since there is a linear increase in protein concentration from 10, 20, 30, 40 to 50 microgram, we should expect a linear plot when quantitating signals from this plot. So the ideal linear response is represented by the dotted line, the gray dotted line on screen here. However, as you can see, none of the housekeeping proteins in HeLa lysate demonstrate that linear response. The reason is signal saturation at protein concentrations in this experiment. So by the way, 20 to 40 microgram of lysate concentrations are pretty standard in Western blotting. So these lysate concentrations are not really out of whack or excessive. So take a look at beta-actin blot and the signal intensity plotted in this chart. As you can see, the signal intensity is pretty much saturated when you go from 10 to 20 microgram lysate concentration. As a result, you mostly get a flat line when you plot those signal intensities, which really means that when you're looking at actin, there is not much of a difference between 10 microgram load or 50 microgram load, which clearly is a problem. GAPDH and alpha tubulin, they fare a little better at 10 and 20 microgram loads, but there is significant signal saturation between 30 and 50 microgram loads, resulting in almost a flat line at higher concentrations. So in summary, signals from housekeeping proteins get saturated with increasing concentration of HeLa cell lysate, resulting in erroneous quantitative results. So the question to ask is, if the use of housekeeping proteins results in signal saturation and erroneous result, which method would provide a linear signal? So the answer is total protein normalization, and the reason is total protein normalization is not dependent on a single protein. I believe that is the reason why the Journal of Biological Chemistry recommends that normalization of signal intensity to total protein loading is preferred. So actually, total protein normalization is beginning to be considered as the gold standard for quantitative Western blotting. The prestigious journal Nature recommends for quantitative comparisons, appropriate reagents, controls, and imaging methods with linear signal ranges should be used. Similarly, Journal of Biological Chemistry, or JBC, recommends that normalizing signal intensity to total protein loading, which can be assessed by staining membranes for total protein whenever possible. Housekeeping proteins should not be used for normalization without evidence that manipulations do not affect expression. Now, it is really not easy to determine in what cell lysate the manipulations that are being used are affecting housekeeping proteins, in which cell lysates the concentrations that are being used in an experiment would result in signal saturation, right? So this recommendation from JBC that you need to ensure that the housekeeping protein works for your experiment means doing a lot of upfront work, doing a lot of validation work to ensure there is no signal saturation and there is no expression level issues for those housekeeping proteins. So that makes it really a challenge to continue using housekeeping proteins, and total protein normalization would be a much easier option because of the concerns expressed by these journals. Now, since total protein normalization is becoming gold standard, we launched No-Stain Protein Labeling Reagent with features that address the current customer pain points. So the No-Stain Protein Labeling Reagent is flexible and works with any protein gel that you are currently using, whether precast or you are pouring your own gel, and any transfer membranes that you might be currently using in the lab. The reagent has an easy-to-use protocol with a labeling time of just 10 minutes. The gel or the membrane labeled with no-stain reagent can be visualized using a wide range of imagers with UV, green or blue LED, or fluorescence light sources. The reagent provides accurate total protein normalization results with broad linear range for detection of 1 to up to 80 microgram of protein loads, which is really a very high protein load for a gel to handle. The reagent is highly sensitive with a detection limit of 20 nanogram per band with a stable signal compatible with downstream chemi and fluorescent antibody detection. So the No-Stain Protein Labeling Reagent, it's a simple kit. It contains 2 bottles of labeling buffers that are stored at room temperature. It has a vial of what we call as an activator and a vial of what is known as a derivatizers. And those 2 vials are stored at -20 C. The protocol is simple mix and add as shown in the schematic. So you start by preparing 1X labeling buffer. You add activator to that. You add a derivatizer, and you basically incubate the membrane for 10 minutes in this solution. There are wash steps before and after labeling to minimize any background during imaging the signal. The No-Stain reagent can be used to stain also the gels or label the gels using a very similar protocol. So it is a pretty simple reagent with a very simple protocol, which gives you quick results, quick and rapid results for your target proteins that can be normalized using total protein normalization. So in the next few slides, I'm going to show you some compelling data on why total protein normalization with No-Stain reagent provides very accurate quantitative Western blotting results compared to housekeeping proteins. So this is the same experiment that I went over in slide 4, right? So it is the HeLa cell lysate with increasing protein concentration ranging from 10 to 50 microgram loaded on the Bolt Bistris Wedgewell gel and transferred using iBlot 2 device on a PVDF membrane. The membrane was labeled with No-Stain Protein Labeling Reagent. This reagent is a label and not a stain because it forms a covalent bond with lysine amino acid, and it has a fluorescent tag with an optimal excitation at 488 nanometer and emission at 590 nanometer wavelengths. So as you can see, No-Stain labeled membrane, it provides a linear signal over the protein load. So from 10 to 50 microgram, you can see a linear increase in signal intensity. The signal intensity was measured and plotted, and it tracks very close to the ideal quantitative response, which is represented by the gray dotted line in this graph. And that indicates that the signal from No-Stain is linear for the protein range used, and it does not saturate. The same No-Stain labeled membrane was then processed for immunodetection of housekeeping proteins, beta-actin, GAPDH, and alpha-tubulin. And as we have seen before, the signal from actin really saturates quite early from 10 to 20 microgram range and continues to be saturated up to 50 microgram, resulting in a flat line shown by the blue line here. GAPDH, slightly better at 10 to 20 microgram range, although still not linear. And at between 30 to 50, there is again a complete saturation. So the dark blue line with triangles indicates that it is still mostly flat at higher protein ranges. Alpha-tubulin, similarly, slightly better response at 10 to 20 microgram, but at higher protein load, there is saturation, which results in getting a flatter line. So as you can see, there is this clear limitation with housekeeping proteins where the protein loads are not properly represented, but with No-Stain labeling reagent, you do get a linear response throughout the range of protein lysate, which really results in a very accurate quantitative Western blotting result. So next, what we did was we did a more numerically quantitative analysis of the densitometric data that was presented before. Now this slide, there is a lot more stat in here, but basically to simplify things, right? The larger the average of percent error, the less accurate is quantitation using that particular method. So in this particular case, you can see actin has the largest error, percent of error, And what that means is that your quantitative results that you get out of actin are the least accurate. GAPDH fares better compared to actin, but again, there is still significantly higher error. And nostain, although the ideal result would be 0.00%, so no percent error at all, but that is usually almost impossible to achieve in experimental world. So nostain, there is error of 4.4%, which is again really low. And what that means is with nostain, you are getting the best quantification data using the protein ranges that were used in the HeLa cell lysates. So in the previous experiments with HeLa cells, right, we did not include a target protein. Now, most researchers are normalizing their target protein with either housekeeping proteins or anything else, but it is always the target protein that is the main interest. So in the next few set of experiments, we have included the target protein. So here, the target protein is AKT1, and this was done with cell lysates from MCF7 cell lysates. Just like in HeLa cells, we have loaded increasing concentration of MCF cell lysates on a bold gel, so from 10 microgram all the way up to 40 microgram. After transferring the protein onto a PVDF membrane, the membrane is labeled with the No-Stain Protein Labeling Reagent. And again, as you can see, the signal from 10 to 40 microgram, it does seem linear and proportional to the protein load. We don't really see a saturation here. And when you plot this, again, which is the green line over here, sorry, the blue line in this particular graph, you are getting a linear signal. Now, AKT1, which is the target protein, when you use the ranges shown here from 10 to 40 microgram, this particular target protein, because expression levels are not as high as housekeeping proteins, you are getting a linear range of expression for AKT1 here, which is shown by this green with the squares on it. So both AKT1 and nostain have a linear signal. But again, if you look at the beta actin or GAPDH signal intensities, and again, this is the same membrane probed with this housekeeping antibodies, you can see the signal saturation happening from 20 all the way to 40 microgram for beta actin, and again, 20 to 40 microgram for GAPDH. So you are getting this sort of a flat line for GAPDH and then beta actin later. But again, if you look at, there is no difference between 20 and 40 microgram load, which is obviously a problem, and that's why results from these housekeeping proteins are not accurate, while results from nostain gives much more accurate quantitation with AKT1 target proteins here. So here again, we looked at the percent error to predict response from this particular experiment. And again, as you can see, the percent error for nostain was significantly lower compared to beta actin and GAPDH, which further highlights the accuracy of nostain-based quantification in MCF7 cell extract as well when using AKT1 as target protein. So if you look at the numbers here, again, as I said, 0.0 would be the perfect result. Nostain is at 5.8%, so slightly higher than the target result. But if you look at actin and GAPDH, they are several-fold higher in terms of the error. So, compared to nostain, they do provide much more erroneous results. So this is again a new set of experiments. So here what we did is we took the Jurkat cell lysate and we looked at a different target protein, cyclophilin B. Again, the results are remarkably similar, that even with increasing protein concentration, no-stain-based total protein normalization provides a linear signal. The target protein, cyclophilin B, also shows a very linear signal, but the housekeeping proteins, beta-actin and GAPDH, are saturated, resulting in a non-linear plot, right? So again, as you can see, the results are fairly obvious. Even if you don't look at the plot, just the image itself shows that no-stain has pretty linear signal, which corresponds to increasing protein loads. But if you look at the housekeeping proteins, beta-actin from 10 to 40, it almost seems there is no difference in protein load. So at 10 microgram, you're almost getting a complete saturation. Same thing is true for GAPDH, right? Even at 10 microgram, which is not that high of the lysate load, you are getting a complete saturation. So really there is no difference between loading 10 microgram or 40 microgram. And the plots, they really represent that. They are almost flat. And that again would give completely erroneous results when you're trying to normalize your cyclophilin B levels, which is really linear at the ranges used here for the jacket cell lysate. Now, the percent error plot again demonstrates that normalization of cyclophilin B with no stain provides accurate results compared to beta-actin and GAPDH, which get saturated with increasing protein loads and result in erroneous quantification of cyclophilin B. Just looking at the values, the no stain percent error value is slightly higher here, 13.5%. So it's the first time we have seen no stain in the double-digit territory, if you will. But beta-actin, it's close to 56%, and GAPDH is at, again, 50%, right? So, there is some error inherent in this particular experiment, but with housekeeping proteins, the error is really so high that it is almost impossible to get accurate quantification results with that. But total protein or TPN is still within acceptable range and would provide an accurate quantification result with cyclophilin B in jacket cell extract. Now, to assess data that might be more representative of the kind of results sought by researchers studying biochemical signaling pathways, we compared a quantification of target proteins from A431 cell extract that had been treated with human EGF, epidermal growth factor, to untreated A431 cell extract. Our findings are illustrated in this figure. As already demonstrated with other cell lines, the no-stain reagent exhibits superior capability for protein normalization compared to the housekeeping protein. In the case of A431 cells, the housekeeping protein beta-actin shows virtually no densitometric signal response to changing protein levels and would therefore not enable any protein normalization in the system. The no-stain densitometric signal intensities, on the other hand, show a linear increase with protein load and track closely with target protein levels in both treated and untreated A431 cell extract. The nonlinear behavior of the HKP signal intensity or signal saturation further confirms with this A431 extracts that accurate protein normalization cannot be achieved with housekeeping protein beta-actin under tested conditions. So again, right, this data set is also very powerful. As you can see, with treated and untreated, you'd see phospho-EGFR levels when they are treated with human epidermal growth factor, but you don't see that when the cells are untreated. And then here's the EGFR levels with increasing lysate concentrations, followed by beta-actin levels. At the bottom is the membrane probed or labeled with the no-stain reagent. Although visually things are pretty clear, but it's always good to look at the graphical representation. that nostain and EGFR, they both have linear signals, right? So, the fact that the target protein in all of these conditions that we have talked about today, and all of the lysate ranges that we have talked about today, the target proteins give a linear signal, right? Which means we are not really overloading the system. We are not really overloading the gels to make beta-actin saturate in this particular case. So we are using the lysate concentrations that are typically used by researchers and making sure that the target proteins are not saturated in those ranges, which is the case in all those experiments. But if you look at signals from housekeeping proteins, they are really saturated pretty early on, right, at 10 microgram loads, which is not that high. So if you look at the expression level of EGFR at 10 microgram, it is quite faint. So if you load less to make sure your actin does not get saturated, you would not see your target protein at all, right? So that becomes a challenge. So the reason that these are the practical loads, because you want to see your target protein first, but at that protein concentrations, your housekeeping proteins are almost always are going to be saturated. And we have shown this with different cell lysates and different target proteins. And obviously we can't include every cell lysate in this particular study, but we feel confident that this is going to be repeated in more lysates when we try and reproduce this set of data. So whether you are labeling or whether you are treating it with EGF or not treating it, you are getting the same results. Linear curves from nostain and your target protein EGFR, whereas saturated data from your housekeeping protein, which in this case is actin. So actin really would not be able to justify quantitative Western blotting with EGFR in this particular cell lysate as well. So again, in this slide, we are looking at the percent error of method calculations, which further demonstrates that in untreated and treated conditions, protein normalization of EGFR and phospho-EGFR can be accurately done with no-stain reagent, but not with housekeeping protein, which in this case was beta-actin. Now, another cool thing about the no-stain reagent is, right, all of the data that we just reviewed, we were labeling the membrane after transfer, right? So it was a membrane, it was working as a membrane label, and that labeled membrane was then used for total protein normalization. But this reagent works really well for labeling proteins in the gels also, right? So here's the data which demonstrates its ability to label protein in the gel. So in this experiment, we loaded increasing protein concentration of cell lysate from 2.5 microgram all the way up to 80 microgram. So again, 80 microgram is a really heavy protein load. And we, after gel electrophoresis, this gel was then labeled using the no-stain protein labeling reagent. And as you can see, even at lower protein concentrations of 2.5 microgram, you are able to see a lot of band pretty distinctively. But at 5 microgram, again, all of the bands are pretty clear. And as the protein load increases, you really don't see a saturation, right? So you do see a proportional increase in signal intensity with labeling of no-stain reagent. And when we plot this in the graph over here, what we are seeing is we are seeing a linear signal with no-stain reagent from 2.5 microgram all the way up to 80 microgram. And it corresponds to this ideal dotted line concentration curve that you can see here. So, the message here really is that the no-stain labeling reagent works really well for labeling proteins in the gels, and it can be linear for a very long range of protein concentration, similar to what we saw with labeling of membranes in the previous data sets. So now this slide really provides the ordering information for no-stain reagent. So the reagent comes in two different sizes, in a regular pack of 40 reaction and a sample pack of 10 reactions. The web link provided here, thermofisher.com/nostain. It has how to use videos for no-stain and then normalize it using the Eyebright Imager. It has an app note on how to use no-stain and Eyebright Imager for total protein normalization. And it also has a web form to request free samples of no-stain. And the website contains additional information about total protein normalization that you might find helpful. So do visit that website and use it for the nice giveaways we have in there, as well as educational material that you would find really helpful. And finally, the Eyebright imaging system is the most ideal for total protein normalization using the no-stain reagent. The Eyebright Imager, which is pictured here, and it looks really nice. It's very functional. It has a built-in no-stain mode that makes it a breeze to capture images of gels and membranes labeled with no-stain reagent, and followed by total protein normalization by using a proprietary normalization software that is included with the imager. So that was my last slide. Thank you very much for attending this webinar today, and I will be happy to take any questions you might have. Thank you.
Thank you, Alok, for your informative presentation. We'll now start the live Q&A session of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. All right, let's go ahead and get started. Your first question is, what are you using for quantitation of signal intensity, and what types of secondary?
Yeah, so that is a great question. So in this particular case, we are using the Eyebright Imager for acquiring the images, as well as normalizing it using the software that the Eyebright Imager has for protein normalization. But there is enough flexibility where you can use any software that you might be currently using for quantitative Western blotting. So for example, ImageJ, which is a free software, also works really well for that purposes, as well as any other software that enable that kind of quantification would work. In terms of the secondary antibodies used here, so we have used the fluorescent secondary antibodies in this experiment. So again, if your imager allows for the ability to multiplex, which Eyebright does, then it offers that option of using fluorescent antibodies for looking at multiple targets within the same blot. We were using the IR and far IR Electrophoresis secondary antibodies because just one channel, which is emission at 488 and excitation at 590. So that one channel is taken by the no-stain signal, but then there are other channels with IR and far IR range, which can be used for secondary antibodies to do multiplexing.
Great, thank you. Okay, this next user says, I have a question regarding the linear increase on slide 4. How precise does the linearity need to be? Would alpha tubulin between 10 and 20 microliters be fine?
Yeah, so I think that's another great question, right? So I think it really depends on your particular experiment. So for example, there are experiments where researchers are comparing differences between a phosphoprotein and a non-phosphoprotein after treatment with an agent that triggers phosphorylation. Now, in those experiments, even a small difference between treated and untreated is going to be extremely critical, right? So for experiments like that, where even a 10 to 15% difference would considered as a very significant result for that particular signaling pathway, it is really important that the signal is really linear, right? As close to perfect as it can get. Because if you are going to have conclusions about 5 to 15% increase in phosphorylation levels as an important signaling event, then you need to be really sure that that increase is real. And that can only be ensured if you have complete linearity with your normalization method, right? So for such experiments, I would say that, the housekeeping protein would not be sufficient. But, there might be other set of experiments where a small variability might be okay, right? So I think it depends on the experiments you're doing. But, to be safe and to make sure that your results are really accurate, it just makes sense to use a method that really gives linear results to begin with.
Is No-Stain compatible with Lycor-itis Odyssey?
So, yeah, I mean, I'm not very familiar with the capabilities of Lycor Odyssey Imager, but No-Stain basically has an, emission and excitation. So, it can be excited at 488 and the emission is at 590. So as long as your imager is capable of doing that, then yes, there would be compatibility with that.
Great, thank you. Now your next question is a two-part question. Is it possible to strip the labeling reagent after probing for another analysis? And can we cut the membrane to evaluate multiple proteins with the same membrane?
Yeah, so, great question again. So with regards to the no-stain labeling reagent, right? So this is a tag that binds to the lysine amino acid covalently. So the way this tag works is you're supposed to probe your membrane before blocking, right? So blocking buffer typically has proteins, so you don't want to label all the proteins in your blocking buffer also. So you have to probe the membrane with no stain before blocking. But once you probe the membrane, there is a covalent bond that forms between the no-stain reagent and the lysine amino acids, which is really stable. And you can then process that membrane for all of your immunodetection experiments. So you can use that membrane and, you can go forward with your blocking steps, primary antibody, secondary antibody, and detection. Whether you're using a chemi or a fluorescent-based methods, it really doesn't matter. This is compatible with all of that downstream workflow, right? So with no stain, you would not have a need to really strip that signal from your protein, because again, it's compatible with everything downstream and would not interfere. As long as you're using the right secondary antibodies, which does not overlap with the signal of no stain. And yes, you can absolutely cut the membrane and probe it with different targets. What you can do is either you can take image of the entire membrane upfront after labeling, or if you want, you can wait till your entire experiment is done, immunodetection followed by the whole detection method, and you can image along with your target protein. So it has that flexibility because the signal is stable and would last throughout your experimental condition.
Thank you. Okay, your next question, how can you use it within an actual Western blot experiment?
Yeah, so that is something that I think I talked about it a couple of times, but just to briefly, go over it again. What you would do is after your transfer is done, no matter what your actual experiment is, after you finish your transfer, you would take that membrane and probe it with no-stain labeling reagent. You can then, after probing, you can take an image right away, or if you want, you can then go forward with your entire experiment of blocking primary antibody, secondary antibody, so on, followed by detection, and then you can take images at the end of the no-stain signal as well as your target protein signals. And the no-stain signal is stable and it will last throughout your experimental process. So you have the flexibility of doing it either ways.
Thank you. Okay, your next question, Alok, does the intensity of no-stain protein labeling reagent change only with the protein load, or are there other factors that can influence this as well?
So yeah, I mean, again, great question again. So the signal that you get from no-stain labeling is really proportional to the protein load, right? So all the data that we showed today, there were ranges from 2.5 microgram all the way to 80 microgram, and you saw that, the signal intensity really represented the protein load in there. So, it is representative of what's being loaded, and it stays linear with increasing protein loads. There can be some factors, like, background with any fluorescent method. There is always a possibility that depending upon how you have labeled and, how you have processed that whole labeling thing, what kind of background you are getting. So, if there is uneven background in the membrane for some reason, which typically does not happen, then there is a possibility that the background can add some variability to your signal. So that is one other method that I can see where there can be some variability through this experiment. But again, the probability of that happening is pretty low if you just follow the steps of the protocol and include the wash steps before and after labeling with no stain. So these are 2 minutes wash with water really. So again, convenient way to process, but it's important that the steps are followed so we don't have any background issues and there are no really variabilities to the actual signal that is coming from labeling of proteins with no stain.
Thank you. Now, how are the signals of protein bands measured with the no-stain gels?
So yeah, great question. I think I answered this earlier on. So in this set of experiments, we are using the Eyebright Imager and the analysis software that comes with Eyebright Imager for quantitating the signal from no-stain reagents. So basically, you select the lane. Every well is going to be a separate lane. So you select that lane, and the software would give you the signal intensity coming out of labeled proteins for that particular lane. So that densitometric signal is then used to normalize the results that were presented here.
Now, after no-stain protein labeling reagent, can I use fluorescent secondary antibodies, red and green, without any problems?
Yeah, so yeah, good question again, right? So as I said, no-stain is a fluorescent probe, which has an excitation at 488 and emission at 590, right? So obviously you would not be able to use a fluorescent secondary antibody that also has an emission at 590. But there is a range of fluorescent secondary antibodies at IR and far IR ranges, with emission at 700, 750, 800, and so on. And all of those can be used for multiplexing along with no stain. So the data set that was presented here, we did use, all of the secondary antibodies were fluorescent secondary antibodies in IR and far IR ranges. So we were multiplexing with fluorescent secondary antibodies in all the data that was presented today. So yes, fluorescent antibodies can be used with no stain. The only thing to keep in mind is make sure you're not using something that has an emission at 590.
thank you. Now, do you have any data from immune cells?
So immune cells is not something, so we did not include any immune cell lysates in the experiments that we have done so far. So yeah, right now I would say, yeah, we don't have any data from the immune cells at this point.
Now, can this no-stain reagent work or be used with proteins made from plants?
Yes, absolutely. So the way the no-stain reagent works is that, it is an FQ label that gets activated when you mix the activator and derivatizer together, right? So it has no signal till you mix the activator and derivatizer together. But once you mix them together, the FQ label gets activated, and that label has a pretty high affinity for the lysine amino acid. So lysine amino acid is really the most abundant amino acid in proteins. So there are hardly any protein that does not contain lysine, whether it's animal or plant or even bacterial. So yeah, it works with all protein types, and the intensity and sensitivity, irrespective of the source of protein, is pretty high with the no-stain labeling reagent.
Now, what if we do not have the Eyebright Imager? Are there any other methods we can use?
Yeah, so the labeling, you can really excite and image it with using a wide range of images, right? So if you have any other imagers that you are currently using, for example, for taking images of either a fluorescently probed labels or even your chemi blots, right? If you have images that are able to excite within the UV range, and then capture the images at 590, all of those images would work, right? So images with UV capabilities, with blue or green LED lights, or of course fluorescent images, that whole wide range of images are compatible with imaging signals from no-stain labeled membrane in gels.
Thanks, Alok. And I do want to remind the audience, any questions that we do not get to today, we will follow up with you via email after the webinar. Okay, your next question. What is exactly measured to normalize the results from a given band? Do you measure the whole lane or the band where the protein of interest is?
Yeah, so for total protein normalization, right? So again, that's a great question. So the total protein normalization, the reason why that method is really linear is that you are not reliant on a single protein. With single protein, there are chances that one protein can show saturation depending upon the expression level. But when we are measuring signal from the entire lane, then that is a method that ensures that the signal stays linear for a wide range of protein load. So in all of the experiments presented today, when we are talking about normalizing the entire load, we are measuring the signal from the entire well and not just a single band. When we are measuring the target protein or housekeeping proteins, that is the only time when we are measuring signal from that specific band and plotting it.
Great, thank you. After using the product, can I do the stripping easily, or are there some specific stripping buffers that should be used?
So yeah, like I said before, right, with this particular reagent, you really don't need to strip the signal. Again, the reason being that it is compatible with the downstream Western application. So we wanted this to be as easy as possible for the current Western workflow. Stripping and reprobing again adds additional multiple steps, and sometimes stripping buffers can be a little harsh, which can kind of compromise immunogenicity of the target proteins as well. So we don't recommend stripping this particular signal for downstream Western applications for those reasons. So, again, if you want to strip, it would work the same way as it works with when you're stripping your antibodies, because antibodies also form a covalent signal with the target protein, and the stripping buffers are harsh enough to break that covalent bond to remove that signal. So if you want to, typical stripping buffers would work, but that's not something that we recommend with this reagent because we feel it just adds extra workload and time for processing of Western blotting, and not really needed with this no-stain reagent.
Thank you. We have time for just a few more questions. Does the probe attach to more than one lysine in a protein?
Yes, so that's a great question also. Every protein has, obviously multiple lysine residues because it is one of the most abundant amino acid within a protein. So in our experience, we feel this reagent binds to about 30% of lysine residues in a protein. So it doesn't bind to every single lysine in a protein, but it does bind to about 30%, which is still a good number of lysine residues to make sure that the signal is strong and the detection limit is pretty high for this reagent.
Great, thank you. Now, how do you go about a no-stain normalization if the transfer has not been even throughout the whole membrane?
So, the question really is that, for some reason, what you loaded on your gels is not evenly transferred onto the membrane, which would indicate that there is a problem with the transfer method or the transfer efficiency, because typically what you load on your gel should be reflected on your membrane as well, right? So that uniformity should be there in your transfer method. So if there is a problem with that, then obviously what the no-stain reagent is going to do, it is just going to detect the amount of protein that is present in every well post-transfer. So if there is a difference in that uniformity, then you are going to see difference in the signal also. But typically, that does not happen during the transfer process. If your transfer has worked well, the amount of protein you load on your gel is going to be reflected on your membrane also. And then the no-stain will just tell you what is the signal strength in every individual well on your membrane.
Thank you so much. Now, can the reagent interfere with the antigen recognition of the target protein?
Yeah, so that's a very good question. So with the way this protein labels work is, obviously they are binding to lysine amino acid specifically. So now if your target protein has lysine in the antibody detection pocket, then there is a chance that there is going to be some interference in detecting that particular target protein. Now, because lysine is so ubiquitous, what we have found is, most antibodies, when they are trying to look for a unique signature, they try to avoid this most common amino acids, right? So most target proteins really don't have lysine in the antibody binding pocket. But again, there are certain targets that we have seen where there would be some interference because of this. Now those are very few and far, but it is really possible that depending upon your target protein, it would be good for you to analyze if there is any kind of interference or not, before adopting it completely for that particular experiment. But what we have seen is that even if there is some interference, all it does is, reduces the signal by a very small margin, which is hard to detect unless you are comparing an experiment where you run, an experiment without no-stain, an experiment with no-stain, and do a side-by-side comparison and quantitate all of it. you might be able to see some difference in for some target proteins. But again, for most target proteins, it's not going to be an issue, and it would really not matter in terms of the results that you're getting with that, because it would uniformly affect the membrane. It's not really specific for any single protein.
Thank you. It looks like we have time for one more question. If my primary antibody detects lysine, will the no-stain interfere with my targets? So would I need to use no-stain at the end?
Yeah, so I mean, that's a great question. So I will answer the second part first, right? So for you to label a membrane, the only time you can label a membrane is before you add your blocking buffer to the membrane, or antibodies also, right? Because this is a probe that detects proteins. So again, blocking buffer has proteins, your antibodies are proteins. So it is going to detect everything if you try to use it after probing it with antibodies and your Western workflow per se. So that cannot be done. This has to be done before blocking buffer step, right? So now if your target protein is identifying lysines only, so antibodies identifying lysines only for that particular target protein, there is a possibility that there would be interference with this. As I said, this probe binds to about 30% of lysines, so not necessarily it would specifically target that antibody binding pocket, but again, because it's binding lysine, there is a probability that you would see some diminished signal from your antibody. So you would have to really test it out in your experimental system and see what kind of interference you're getting, and is that interference significant enough to block the signal to a level where your signal is really low, and then you can't use this reagent, right? So that is something that you would have to test it out.
Thank you so much, Alok. Do you have any final comments for our audience before we go today?
Yeah, so very exciting questions from the audience. Great interest. So just glad to see that there is interest about saturation of housekeeping proteins, which is really the most commonly used method right now. And all I would like to say finally is that nostain really offers a great alternative to housekeeping proteins. It is simpler, faster, and accurate, right? So if you don't probe for housekeeping proteins, you're saving all of those antibodies, you're saving all of those steps that are required to probe the target protein and then process them. So this is really a great alternative. It simplifies your workflow and enables you an ability to get very accurate quantitative Western blotting results. So I would say, if you are interested, please go to thermofisher.com/nostain webpage, where you can order, samples, free samples. We'll be happy to send it out to you so that you can test it with your target proteins and experimental system, and you can see for yourself, how this is going to make your Western blotting results much more accurate and also convenient at the end of the day. So I would like to thank you once again for all the exciting questions and listening to the webinar today, and hopefully this helps you with your experiments within the lab.
Thank you, Alok, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their questions. Again, any questions we did not have time for today and those submitted during the on-demand period will be addressed by Alok via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.